delighted to introduce Dr. Brenda Rapp, who is a professor and the past chair of the Cognitive Science Department at Johns Hopkins. And she's a world-recognized cognitive neuropsychologist whose work has focused on the mechanisms and neuroanatomy of written and spoken language. I've known Brenda for, I guess, since the 1990s, longer than at least I would like to admit. When, as a young scientist, she was already very highly regarded for her work on semantics and lexical processing. Brenda, you may be just like a couple of years ahead of me, but when I was a postdoc, I remember reading seminal early papers by Brenda and her mentor, Alfonso Karamazza, that really changed the face of the field. And she's continued to do work of that caliber. She is the editor-in-chief of Cognitive Neuropsychology and is on the editorial board of Language, Cognition, and Neuroscience, and the associate editor of Frontiers in Psychology. She's published over 120 articles and given many keynote lectures at international conferences. And today she's going to talk to us about understanding language recovery at multiple network scales, from voxels to hemispheres. So welcome, Brenda, and you should be able to share your screen. Let me know if you can't.
Great. Thanks so much for that introduction. That was great. And it's so amazing to see all these people here that I haven't seen for a while. You know, we've all been kind of deprived of people. And so it's just a treat. Let me figure out, though, how to do what I need to do. The technology, though, is always the little stressful component, right?
For us of a certain age.
Exactly, right? Okay, so let's see. I need to, let's see. Oh, wait. I need to, sorry. I want to share my screen, but I also want to share the sound. Okay, yeah, I have that. Which one is it? Let's see if that's the right one. Okay, can you guys see that?
Yes.
Is that good? Okay. And so, Laurel, I think we talked about, I'm happy to take questions, like clarification questions, not so much discussion questions, just because, you know, otherwise we can get really distracted, I especially. But clarification questions would be great, actually, because I don't want people to feel like they're lost, you know, the whole talk, and then it would have just been a waste of time. I don't know how you'll handle those, Laurel. Are we doing questions like in chat, or are people raising hands, or what do you want to do?
Whatever you prefer, Brenda.
I won't be able to, I know I won't be able to monitor anything while I'm talking.
No,
so
okay. So
if you want to monitor, that's fine.
I'll monitor the chat box, and I think that might be a good way to do it. If you have a question for Brenda, just write it in the chat box, and then I will alert Brenda, just so that you don't get a million questions. We'll go through, I'll screen them a little bit, just make sure if they're clarification, I'll interrupt you. Yeah. Okay, great.
That's great. Okay. That would be great. Yeah, just interrupt me with clarification questions. Okay. Not a problem, really. Okay. So I haven't given this talk before, so we'll see how it goes. And Laurel said, oh, maybe you could talk about, you know, representational differentiation, or maybe you could talk about, you know, the stroke phenotype. And I said, oh, I'll talk about both of those. So I'm going to be trying to combine a number of things into one talk. And so hopefully this will all work while still trying to, you know, not go into too much detail. So it's hard to know exactly how much time this will take, but I'll do my best. Okay, so let me get started. And first I want to acknowledge the two people with whom I collaborated with on this project, Yuan Tao and Jeremy Purcell. They really, you know, deserve all the credit for the work that was done here, and they're just great. And I think Yuan is here, I hope. So she will also be available to answer some questions. Okay, so let's get started. So let's take a look at what it means what it looks like when someone is recovering or has improved their performance as a result of treatment. So let me start off with this video of a gentleman who was involved in one of our dysgraphia treatment studies. And what you'll see from him, what you'll see from this video is his attempt to spell the word emperor at different points in time over like about a 3-month period. So each one is like is an attempt during this 3-month period, and in between these attempts, he was receiving treatment, and this is one of his trained words. Okay?
Emperor. Emperor.
Another time. Another time.
Not good, but.
Okay, so you can see how effortful and painstaking, but yet you can still see the progress, right? So over the course of these 3 months, this word and many other words, improved in this way. And so this is, just a recap of that. And so the question is, the question that we're interested in was, well, what is changing in the brain? What neural changes are supporting this kind of recovery? And so what you have here simply is just that we have a number of individuals who underwent therapy of this, and dysgraphia therapy, and you can see that they all improved. They all improved significantly from pre to post treatment, and they maintained their results for the most part. I mean, there was a little decline, but they were still largely maintained. And the other thing to note is that they not only improved in the trained words, the words that they trained on, but they also improved on untrained words. So there was generalization to untrained words. But interestingly, and I think importantly for the results that we're going to try to interpret, is that they didn't improve across the board. So they improved, in spelling, they improved in their trained words, they improved in untrained words, but across all the other tasks, at least that we evaluated them on, they didn't improve. Okay, so this is really a selective improvement, and that's going to, I think, help us in trying to interpret some of the brain changes that we're going to observe. So the roadmap for today is I'm going to give you a little bit of an introduction on networks, and then I'm going to talk about recovery of networks at these 4 different levels: at the micronetwork level, within network connectivity, across network connectivity, and interhemispheric connectivity. So the more standard approach that I've carried out, and many of you have also carried out in terms of trying to understand recovery, involves doing fMRI, a task-based fMRI, for example, and then localizing, where's the brain activity changing from before to after treatment. So that's a very standard approach and very useful. Okay, and that approach, has allowed us to investigate questions like, are changes perilesional? Do changes involve homologs? Are there other kinds of changes? And they yield results like the following. So this actually is our mean bold data from this study where we can see that from pre to post treatment, there are these areas where the mean activity of the brain has changed from pre to post treatment. Okay, so that's one approach, but that's not the approach that I'm going to be talking about today. Okay, so another approach, not there's anything wrong with that, but that's one way of looking at the brain. And so another approach that we've been exploring for the last few years involves looking at networks, network properties, and connectivity between brain areas. So instead of looking at changes in brain areas, it's looking at the relationships in the activation patterns between and across brain areas. And there are many ways that, many aspects of connectivity that one can look at, many characteristics of networks that one can look at. This is a list of some of the network measures that, you know, people you can evaluate a network according to these measures. We're not going to talk about all these, but just to give you an idea of sort of the richness of this approach. And this is a standard toolbox that's used for that. So that's the approach that we're going to be talking about. And so it's worth, you know, clarifying what we mean by a network. And so a network is typically characterized as having nodes, which are thought to carry out some kind of computation, and connections, which are sometimes called edges, which involve, you know, information transfer between these nodes. So just a sort of a simplistic way of thinking about a network is there are nodes and edges. Nodes carry out computations, and information from nodes is communicated to other nodes via connections. There are other ways of thinking about it, but that's certainly a good starting point. And you know, in the literature, we can evaluate structural and functional networks. So structural networks would be the white matter connectivity between brain areas. Is that or is that just a microphone? Sorry. Okay, I'll go on.
I don't know. It kind of looks like glass.
Can everyone mute themselves? Hello, everyone. Please mute yourself. Thank you. Oh, I should be able to do it.
You can probably do that. Yeah. Okay. So there are structural networks that, you know, where the focus is on the white matter connectivity between brain areas, and functional networks, which is going to be the focus of what I'm going to be talking about. And when we look at functional networks, what we're looking at in terms of the connectivity between nodes is we're looking at the degree to which different nodes or different brain areas, their activity is synchronized. So
sorry, I'm doing it.
Definitely feels better.
So from a functional point of view, two nodes would be strongly connected if their activity is highly synchronized. So, you know, we know what a structural connection means. You know, you have white matter connecting two areas, but a functional connection is not a physical thing that we're measuring. Rather, instead, we're measuring the degree to which two areas, the activity of two areas is synchronized. So, it's, you know, the activity is going up and down, you know, in a synchronous way, the extent to which it's doing that. Okay. And key concepts in thinking about networks, really sort of the fundamental concepts in thinking about networks, are segregation and integration. And the idea is that if you have nodes that are locally very integrated, so they're communicating highly with one another, that creates a subnetwork, if you wish, or a network of nodes where you have a lot of local connectivity, a lot of local communication. We could call that a network, a subnetwork, we could call it a module, we could call it a component. All these different words are used really to refer to the same idea of this highly interconnected set of regions. And this strong local integration or interconnection has been assumed to be important for specialization. So nodes that are specialized to do something, they're able to do that efficiently and automatically when they're locally very interconnected. And another advantage, presumably, of a system that has multiple highly interconnected modules is that if you damage one part of the system, that area is damaged, but it doesn't necessarily have to spread throughout the system. So these are just some of the properties that local integration provides, or some of the properties that it creates and some of the advantages that it provides. But we can not only talk about local integration or the degree to which nodes are segregated from other nodes amongst themselves, but we can also talk about, and we can measure the degree to which nodes communicate globally, are connected with many other nodes across the network. So that you can have broad communication across the network, and that is, you know, described as local integration. So both, and the advantage and the usefulness of global integration is it allows for coordination across many different modules. So you can have these segregated modules that are segregated unto themselves, but they have they have some communication with each other or with other modules, and that would be their global integration. And that allows for this communication across modules. For complex tasks, it allows for coordination, and it may allow for, when we're faced with novel tasks that we haven't performed before, allows for some kind of flexibility. The main idea here is just that the trying to evaluate the degree to which nodes and their connections are locally connected to one another and globally connected to other nodes are sort of the basic parameters and principles of interest, really. Everything else kind of boils down to this in network analysis. Hopefully that made some kind of sense. Okay, but the question, and maybe you're thinking about this, is okay, so you said networks, you know, they're nodes and they're connected, but what counts as a node? And so there are nodes at many different scales. So, you know, you know, of course, neurons could be nodes, but if we look, you know, in terms of the instruments that we have in terms of fMRI, then we could think of individual voxels as being nodes, and we could be interested in their connectivity with other voxels, or you could have, you know, brain regions that are nodes in a larger network, or you could have lobes that are, you know, the nodes of a larger network, and you can have hemispheres that are, you know, nodes to each other. And so the scale of nodes varies, and therefore the scale of networks varies. Now, the question about how integrated and segregated networks are relative to each other, therefore, can be asked at all these different levels. We can ask this about networks of voxels. We can ask about networks of brain regions. We can ask about it for networks of larger brain regions. So networks can be, you know, conceptualized and evaluated at all these different levels. Okay, and you know, it's been argued by many people that the brain, you know, is executing computations at many different levels. So the brain isn't just computing at one level. There isn't sort of a privileged level at which all computations occur and everything else is, you know, just kind of supporting structure. And so the basic idea is that, you know, the brain exhibits this kind of hierarchical structure where you have smaller networks that form part of larger networks, which in turn form part of even larger networks. And computations are occurring at each of these various scales. Okay, and so the question that we've been looking at over the last few years is, you know, how are computations at these different network scales? How are they affected by intervention and recovery? That's the question that we've been interested in. And so the data that I'm going to be describing to you today comes from a study that we carried out that, depending on which particular set of results, it involves between 15 and 20 individuals. They all suffered single left hemisphere chronic strokes, they're very chronic. They all exhibited acquired dysgraphia. They may have other disorders as well, but they all did have acquired dysgraphia. They're on the well-educated, on the higher educated end of things, so between 12 and 19 years. And we also have some neurotypical age match control subjects that we'll be hearing about. This is an overlay of their lesions, the lesions of this group. And you can see that, you know, typical of middle cerebral artery infarcts, you know, you have this sort of concentration of lesion in the insula. And I'm highlighting this area here with a circle for, as you'll see, it becomes prominent, you know, in the results that we have. But it's an undamaged region. All the participants are, you know, not damaged in this region. And so, I mean, there are other regions where very few are damaged, but, you know, everybody has this region as an undamaged region. And this is a ventral part of the inferior temporal gyrus and the fusiform gyrus. The study, you know, had the following structure. We did a bunch of assessments. We did some neuroimaging. We did about 12 weeks of behavioral intervention. Then we did another, you know, whole series of behavioral and neuroimaging assessments. And then we also did a follow-up beyond that. And the data that I'll be talking about come from this study. In terms of the treatment they had, we developed individually tailored stimulus lists for people. So they were working on words that were selected for them. There were words that each person had difficulty with. We used a very basic spell, study, spell remediation approach. I think that was first, hey, Paige, that was first introduced maybe by Paige Beeson, who's here, and that we've used extensively in the lab since then. And this approach targets really the central components of the spelling process. So we're not, you know, focusing on the motor aspects of spelling, but, you know, the cognitive central aspects of spelling. And they engaged in treatment, usually two sessions a week, and on, you know, for about, like I said, about 3 months. So on average, 26 sessions. These individuals were scanned, as we said. In the scanner, the data, I think all the data I'm going to be talking about today involves a spelling task that they did in the scanner. And I can talk about that later if anybody's interested in knowing what that spelling task was like, but they did a spelling task in the scanner, and they had 2 sessions of scanning before and 2 sessions of scanning after their treatment. Control participants did the same kind of spelling task, but they were only assessed at 1 time point. Okay, so we're going to be looking, like I said, at functional connectivity, which is looking at the degree to which brain areas show synchronized activity, some version of that, and across these network nodes at different levels. Usually this is done using resting state fMRI data, but that's not what we used here. So we used what is sometimes called background connectivity data for those who are interested. This involves taking the task-based bold response, because I told you that the participants did a spelling task in the scanner. We take the time course of the brain response for the spelling task, and we actually take out all the components that are associated with the spelling task itself, and what's left is what's called the residual. So we've taken out the spelling components of that brain response, and we're left with a residual. There's a residual time course. The amount of residual bold there is, at every time point creates a time course, and one could talk about why that's a useful approach or not, but other people have looked at this and said that it, found that it basically reflects what you see in resting state, but it also has a task-based element to it. So it has some component of the task still remains in this measure. Okay. And the last thing for the introduction that I just wanted to lay out there, regarding spelling, people have done a lot of work trying to understand the cognitive components of spelling, and we don't need to go into them today, but just to remind you that spelling is a complex cognitive task that involves, a number of different components, and that those components presumably are instantiated in the brain areas that we find that are reliably activated when people do spelling tasks in the scanner. And this is a snapshot of the brain areas that are most reliably involved in spelling tasks. And again, I'll highlight this ventral temporal occipital area. It's one of many, but again, it's that area that we saw undamaged in the across our group. Let's see, do I want to say anything else there? Yeah, I guess I could also say that in terms of, how these functions map onto these brain areas, again, focusing in on this ventral occipital temporal area, it's an area that, for those of you familiar with reading literature, it's also referred to as the VWFA. So it's been associated with reading, and in spelling, the work that we've done strongly associates it with lexical orthographic representation and processing. So in terms of like the orthographic lexicon, if that's what words that you're familiar with, or the storage and processing of our long-term memories of the spellings of words. We know we've stored in memory how words are spelled. That's how we can, spell words. And this area, not the only area, but this area has been strongly associated with the storage and processing of those long-term memories that we have of our knowledge of word spellings. Okay, so let's start to look at, we saw this improvement in spelling performance, and I want to start with looking at what we could call a micro network recovery. Okay, so what's happening down at the voxel level. Okay, and so step back just for a little bit, there's the question of, well, is there a neural signature of learning? So, what does learning look like in the brain, at the voxel level? And because that might be a basis for thinking about what does relearning, look like in the brain. And so one of the things that, in terms of trying to understand this neural signature of learning, one of the things that seems to be the case is that neural representations are spatially distributed. And what does that mean? That means that our knowledge, let's say, of the spellings of words, if that's, I'm trying to find my cursor here. Where is my cursor? Here it is. The knowledge, let's say, of the spellings that we have of different words, knowledge of spelling of any given word may be distributed over multiple voxels. Okay, so your knowledge of the spelling of the word knowledge may be distributed over a number of different voxels in the brain, as then would be your knowledge of the word, you know, table or the word telephone and so on. So these would be distributed. The idea is that not just for spelling, but for many things, knowledge seems to be spatially distributed across neural tissue. And we're, you know, when we're working with fMRI, we're talking about the scale of voxels here. Okay, so that's sort of, you know, that's a pretty standard assumption, not that it can't be questioned. And one of the things that people have shown doing electrophysiological work is that, that with learning, neural representations, so it's like a separate result, neural representations become sparser. So if neural representations are distributed across neural tissue, any given representation, what people have shown even with single cell recording is that with learning, the overlap across neural areas for any given representation becomes less. So they become more differentiated. So if two representations produce very similar responses across across neural tissue, so maybe, you know, when you don't know, let's say, a word very well, the response in a brain area, you know, for the one word, maybe not that different from that brain area's response to another word. They're overlapping. But as you learn whatever it is, in this case words, but it doesn't have to be words, as learning happens, those representations become less overlapping. They're less similar to each other, and therefore the neural responses to each of them become more different. At first, when they were overlapping, if you could take a snapshot of that neural response for the two of them, they would look kind of similar. You know, they'd have some differences, but they look kind of similar. But as learning improves, those neural representations become more more differentiated from one another, and they're more different from one another. So if you were to think about it in terms of how correlated their patterns are, they become less correlated, right? They become more differentiated. So that the representations in an area where you have a lot good learning, those neural response patterns would be more different, so more heterogeneous. The response of that area, when you look at it, you know, in response to, let's say, many different words, when you don't know those words very well, then the response of that area is going to look really similar for all those words. As you learn those words, the response of that area is going to become more differentiated. It's going to be more heterogeneous because those patterns are going to pull apart from each other. Hopefully that made sense. Okay, so increased differentiation can be thought of as a marker of learning. And so this idea was put to the test in fMRI by Jiang and colleagues, where they actually quantified how different the responses were in adjacent voxels, how different they were to each other when these individuals, when autistic and healthy individuals were looking at faces. They were looking at the face region of the brain, and they were seeing how different are the responses. Show a face, what are the responses? Show another face, what are the responses? And looking at how different those neural responses were. And they found that the autistic individuals, the responses were less differentiated from one another, right? So they were less heterogeneous, they were less different. And the degree to which they were less different was associated with their difficulties with processing faces. And so that, you know, makes sense, because their face representations, you know, may not be as strong, they might not be as well learned, if you wish, and so they're less differentiated from one another. Building on this, on these results, Jeremy Purcell created an analysis which he calls local heterogeneity regression analysis. And we don't need to go into any of the details here, but so I know some people might be interested in at least understanding a little bit of what we did. So basically the way this works is you get a set of adjacent voxels, which is like a searchlight. So you could have let's say 7 voxels, and you record you have the time course of the response of each of these voxels. And basically what you're trying to understand is how similar is the time course of all these voxels that are next to each other to one another. Are they really similar to one another, or are they more different from one another? If they're more different, that supports the idea that whatever it is they're representing, you know, is pretty well learned. If they're all kind of similar to each other, then what those voxels are representing may not be as well learned. Okay, that's basically the idea behind it. And it's done via regression analysis, where you take one time course and you try to predict the other time courses. But the bottom line is you're just trying to understand how different the responses are. And you look at this, you go through the whole brain looking at little clumps of voxels, so that you can evaluate for every voxel how similar its response is to the voxels around it. for the brain, for the brain activity that you've collected. Remember, we're collecting this brain activity while people are doing a spelling task. Okay, so that's the brain activity. We're looking all through the brain to see for every little clump of voxels, how similar are the responses. Well, that's what we will be doing. How similar are the responses to one another? Sorry, this is not the spelling data yet, but that's the idea behind it. Okay. So to see whether or not this made sense, I mean, people have shown it for the faces and, there's electrophysiological work, but does it make sense for the kinds of things that we're interested in? What Jeremy did was he said, well, before we get to patients, let's just start with healthy controls. Let's look at reading, just flashing words. People are reading words. And if this is, if this makes any kind of sense, then what we should expect to see is that if we compare these local heterogeneity for high frequency words that people are seeing versus low frequency words or pseudo words, if this is really indexing learning, then we should see more heterogeneous responses, right? There should be more differentiation for high frequency words than for low frequency words or for pseudo words, right? The better learned high frequency words, the responses should look more different from each other. Yes, Laurel.
Oh, sorry, Brenda. We had a question from Sharon, who I belatedly saw it. I'm not a very good monitor, apparently, but Sharon asked whether you used a lexical or a sublexical approach, because it would help her think about linking up the training with the activations,
yeah.
with the
okay, yeah. So the
training. I think you mostly answered it, Brenda, but yeah. Thank you.
Yeah, the training was a lexical approach.
Right.
Yeah, it was.
Yep. Thank you.
Yeah, no problem. So in this study, before we've gotten to our dysgraphia stuff, we wanted to see whether this idea of whether we could really look at this heterogeneity as an index of learning. And so we were looking at it to see whether or not it indexes high versus low frequency or pseudo words. Okay. And so we subject in the scanner just, passively viewed high, low frequency words and pseudo words. And we went through the brain, trying to see for every group of voxels how different their responses were to high frequency words, low frequency words, and pseudo words. And this is what we found. The only area in the brain that showed a difference in terms of local heterogeneity that varied according to word frequency was, it's kind of tilted here, but it's this ventral occipital temporal area, the VWFA area, is showing this, so we're not looking at activation levels, remember guys, so this is not they're more active for high frequency words or less active. We're not looking at that. One could look at that. What we're looking at is how similar are the local responses to each other when they're seeing high frequency words. So, locally the responses are more different from each other for high frequency words than for low frequency words. They're overlapping less for the patterns across voxels are overlapping less for high frequency words than for low frequency words. So then we thought, okay, now that we have some kind of confidence that this may actually be indexing learning and orthography, we took our participants before their treatment. We had their data from the spelling task. We're looking at the background connectivity. We did the same thing. We did a searchlight across the whole brain, trying to see, are there areas that now not high versus low frequency words, but that are sensitive to how accurately these participants spelled these words, not how accurately they spelled them in the scanner, but how accurately they spell words outside the scanner. Like, what is their dysgraphia severity like? Okay, so we're looking to see whether there's a part of the brain that seems sensitive to dysgraphia severity. Okay, got it? Okay, and this, I mean, I have to tell you, we were shocked, but this is the only part of the brain that we found that showed this differentiation, in the bold response during this. They were doing a spelling task. So they were doing a spelling task, but what we're looking for is the degree to which that differentiation during a spelling task is correlated with their dysgraphia severity measured outside the scanner. Oh my God, I gotta do something with this phone. Actually turn it off. Okay, sorry guys. And so you can see that this is we're looking now at a basal view of the brain, but it is the ventral occipital temporal area, again, the VWFA, and you can see the correlation between the degree of differentiation, the degree to which these voxels are responding differently is higher. So they're responding more differently for individuals who have less impairment in spelling. So these responses are more differentiated when you have less severe spelling deficit, presumably because your orthographic representations are more differentiated from each other, and hence you're able to spell better. Okay. And so that was good. And then we were also curious to know, so these people, remember, everybody has this brain area intact. So it's not because they have damage there. This is really reflecting the integrity of their spelling system. We were curious to see whether, even before we started training, does the degree to which you have this heterogeneity of response here, does it predict how well you're going to respond to treatment? You know, covarying out your pre-treatment severity. And we found that indeed it does. So people who showed a greater heterogeneity of response, which is associated with less severity, they also responded better to treatment. So that's all before treatment. And then we wanted to know, well, what, you know, we saw this guy change, you know, we saw this gentleman and we saw him improve, you know, presumably his cognitive representations are becoming more differentiated, that's what we're seeing as he improves his spelling of emperor. And so we in fact find, when we look across the brain, this is the area where we see change in heterogeneity from before to after treatment. Okay, so this is a brain area where the pattern of activities in these voxels is changing from before to after treatment. Okay, so when we put those three things together, what we can see is a sort of area in these three analyses that, you know, indexes severity of impairment, predicts response to treatment, and is changing as a result of treatment. And so why this area, right, this ventral occipital temporal cortex, okay, so as I said, it is an unlesioned component of the healthy spelling system in all these individuals. And we looked across the whole brain. I mean, other areas could have, you know, come up, you know, but they didn't. I mean, there are other areas that are, you know, that, I mean, we looked at the whole brain. Not every area is unlesioned in every participant, but there's plenty of unlesioned cortex in lots of people, okay? So this is an unlesioned component of the spelling system, and that may be part of why it is showing this response. So that's that. And as I told you, it's critical for the storage. From other work, we know that it's critical for the storage and retrieval of orthographic long-term memory representations, our knowledge of the spellings of words. So it makes sense, sorry, that's just those results, but it makes sense that if what we're improving are these representations of people's knowledge of the spellings of words, it at least makes sense. It's plausible that this is a brain area that would be sensitive to that kind of improvement that we're seeing. Okay, so let me summarize these micronetwork changes that we've seen. So what, you know, sort of the take home from this, even if, you know, the methods aren't, you know, interesting, or, you know, if it was too fast. So what we found is that treatment can produce these, you know, representational changes at the micro network level. We're looking at these little mini networks, the relationships amongst, you know, voxels. So we're really looking at the millimeter scale here. Okay, so we've presented evidence that treatment can produce changes at this level, and that in dysgraphia, at least, these changes are localized to this undamaged ventral occipital temporal cortex. And our interpretation, as I've already suggested, is that relearning involves, at least, you know, in dysgraphia, and given this kind of training, relearning can involve, and in these cases, we think it did involve the strengthening of orthographic representations, or maybe their retrieval. I mean, it's difficult to, you know exactly what is happening with these representations, and that this strengthening is reflected in what we've called as redifferentiating of the neural representation. So the neural representations, when the system was damaged, even though this part of the system wasn't damaged, but when the spelling system was otherwise damaged, we would argue that these representations became less differentiated, and with training, we can see a neural response that seems to indicate that there's been this redifferentiation. It makes sense cognitively, and what we're seeing is a neural signature of that. Okay, so part 2. So that's the micro level. And so we're going to kind of move up a level and look at sort of the within network connectivity at a kind of higher scale. I maybe could have called that something else. Hard to figure out terms for all these things. But hopefully, if you don't understand, you're going to tell Laurel. Okay, so so we're now looking sort of at a higher level, not voxel to voxel relationships, but we're looking at now relationships between nodes in sort of clusters of nodes. Okay, so that's what I mean by kind of up at a higher level. And to do this, we take another approach, and I think all of the remaining sets of results involve using these functional connectomes. Yeah, I think they all do. And so what do we mean by this? Okay, it's not really that complicated. So I just want to make sure that it's clear. So like I said, a network, is a bunch of nodes, and their, and their relationship between, each other. And so you have to decide on your nodes. You could, and so what we did was we used a set of nodes, which are they're 5 millimeter. I think they're 5 millimeter spheres. Yuan can correct me later. I think they're 5 millimeter spheres of voxels, and so we used 235 of them that that Power et al. Had said were like good nodes. I mean, that they sort of have good coverage. And so what we did was we looked at these 235 5 millimeter diameter, I think, nodes, 5 millimeters, and so they have a number of voxels within them, right? So for every node, it's going to have one value. We're going to look at the activation time course. Remember, we have this time course that we're looking at. We're going to get an average time course for all the voxels in each node, and we have 235 nodes that are going to make up the universe of nodes that we're going to be looking at. Okay, so we have those nodes, and we're going to be interested in the connectivity between them. That's where we start. And so what is a connectome? The connectome is just, if we take our 235 nodes and we look at how synchronous the activity is between every pair of nodes. So we have 235 nodes, and we compute how synchronous the activity is between every one of those nodes and every other one of the other nodes. And so we get a value. There's a correlation value that indicates how synchronously every pair of nodes is firing relative to one another. And we can take all those correlation values, and that's our 235 by 235 correlation values. Yeah, that shows you like two nodes and their correlation. We can take all of them and just put them in a matrix. All the matrix is a matrix of 235 by 235 correlation values. To each of those correlation values, we can assign a color, just because it's hard to look at that many numbers. And so you can create a correlation matrix where each cell, which is just indexing how synchronously those two nodes are firing, you can just give them colors where the orange, yellow to orange is high positive correlations. And that's really all a connectome is, a functional connectome is. It's just the correlation values between all the pairs of nodes that you've decided are going to be the pairs of nodes that you're going to look at. And so for the analyses that we're going to, and so there's a lot of things you can do on this, and ways you can analyze this connectome. What we did for our analyses is that we wanted to, for various reasons, have kind of, we needed to get some organization to this connectome. So you can even see by looking at it that there's some, nodes that seem to have high correlations with each other. That's the yellow parts. They kind of cluster together. They're really highly correlated with each other. Those are the yellow things, and, other ones are less correlated. So they clearly clump. Okay. And so these clumps are what can correspond to networks, some people call them, or modules or components. Okay, so there's all these terms that people use for them. We're calling them modules or components. Sometimes we use the word network too. Okay, but this is really what it is. It's sort of nodes whose activation correlates with one another. They form little clusters. Okay, and so in order to figure out of this whole connectome, like what are the basic clusters? Like what is the overall organization of the whole? Remember, this is across the whole brain, these 235 nodes. How's it organized? What we did is we got a connectome for each of our control subjects. We averaged them together. So now we have the average relationship between every pair of nodes for our healthy control subjects. And we do some hierarchical clustering to see like how do they group? I mean, we can see that there's grouping, but how do they group into how many groups are there of nodes? And so we identified 10 clusters or modules or groups of nodes, okay, and we could talk about how that was done, but the basic idea is, is really straightforward. These are just nodes whose activity is correlated with one another, okay, and so we identified 10, and these are the names that we assigned to the 10: a prefrontal module, perisylvian, ventral frontal parietal, dorsal frontal parietal, medial occipital, and so on. So these are the 10 prominent groupings of nodes, and we're going to call each of these modules, like I said, we also call them networks. Okay, they're networks within this larger network. Okay, and so once we have this, once we've decided what our modules are, we can then ask about, well, the nodes that are with, remember, a module is a bunch of different nodes that are, somewhat correlated with one another. Then we can ask things like, well, how correlated are they with one another? We can look at the within module connectivity. I mean, we know that there's some there because that's why they're a module, but how correlated, are they? And does that change with treatment? Does it change with damage, and so on and so forth, right? So we know from healthy controls that these 10 groups of nodes, kind of cluster together in healthy controls. And then we can start to ask questions about, how this changes, with brain damage and recovery. Does the within module connections change? Do the connections across modules, do they change? All this information, is in this connectome. We just need to extract it and, analyze it. And so we have our reference module, our reference structure with these 10 modules, and then we can take all of our patient connectomes. We create one for each patient, right? We have all the 235 nodes. We see how correlated they are. We create, each patient has their own connectome. We have a connectome from before treatment, and we have a connectome from after treatment. We can see how those connectomes change from before to after treatment. We can see how they're different from patients to subjects. We can see how the internal organization of those connectomes is affected. For the patients, the nodes that are lesioned are not part of their connectome. So the patients have little gaps in their connectome where they have lesions. Okay, so there aren't connections, because if you don't have a node, you can't look at its response pattern. You can't correlate it with anything else, right? So the damaged areas, those nodes that are in damaged areas are removed from the analysis. Okay. Okay, so our one of the first question that we're going to look at is, so we have these 10 modules that I talked to you about, and so we asked, one of the questions we asked we asked whether the within module connectivity, so there's the 10 modules that are in the normal healthy brain, does this within module connectivity change with treatment? That's the question that we had here, okay? And to do that, we compared the amount of within module connectivity, the degree to which nodes within a cluster, right? Remember those 10 clusters that I showed you, the degree to which nodes within each of those clusters are connected to each other before treatment, and the degree to which they're connected to each other after treatment, okay? And we can average that across all 10 of those modules, the 10 modules that I showed you. And what we found was the following, okay? So let me, first of all, just over here on the right, we actually had 2 control groups, because one group we used to identify those 10 modules, and another group we used to check and make sure that the results that we got were consistent even for a group of control subjects that weren't used to identify the 10 reference modules. And you can see that they're very similar to each other in terms, they're not different from each other in terms of how interconnected those 10 modules are on average. Okay, so that's just the control subjects. They're very similar to each other, which is comforting. And so here are our patients. The average amount of connectivity within those modules, those 10 modules that I showed you before treatment and after treatment, okay? And so first of all, you can see that both before and after treatment, they have lower connectivity. So the synchronicity of their activation within modules is lower in the patients than in the controls. That's just the overall result. But in terms of treatment, what we see is that the amount, on average, the amount of internal connectivity within these modules is increasing from pre to post treatment. Okay, so internally, on average, these modules are becoming more tightly internally connected. That's what that result shows us. Okay. And so then we asked, well, is that true? This is an average result. Is it true for all of these 10 modules, right? Remember I told you there's these 10 modules. We're looking at their internal connectivity. On average, it increases, but is that true for all of them? And it turns out that it's not true for all of them. And what you see in this histogram is the pre and post treatment for each of them. See that here are the 10 modules. Colors are crazy, and sorry, and they're going to change from study to study. But anyways, so here are our 10 clusters, or our 10 modules. And so that, because looking at colors is not very, you won't remember which one goes where, but so this is here to help you understand where these clusters are in the brain. And these are the pre-post treatment within module connectivity values. How connected are those nodes within a module to each other before treatment and then after treatment? And the only cluster that shows a significant increase, there's many that show numerical increases, some that don't show any changes, but the only cluster that shows a statistically significant increase from pre to post treatment is the ventral occipital temporal cluster, this brain area that we saw in our previous set of results, where we saw this, remember the local heterogeneity down in the voxel level it was changing. Now we've really gone up to a completely different level. We're talking about the, the synchronous activity between nodes. Each of those nodes, is 5 millimeters, it contains a number of different voxels. And within this larger region, what we find is that the connectivity between at this much higher scale, right? Not voxel to voxel, but across, we're looking now at centimeters, or a couple of centimeters, several centimeters. You see that this connectivity now has increased from pre to post treatment. And so this is
sorry, Brenda. We have
yeah.
a clarification question. Go ahead, Umi.
Hi, Dr. Rapp. I just had
Hi.
a question. I know you said you're using modules and components and networks interchangeably. I just wanted clarification on, did you do a modularity analysis with those nodes, or are they the results of the hierarchical cluster?
They're the result of the hierarchical clustering.
Okay.
Yeah.
And I know Power they've split their nodes, I think, through a modularity into different networks itself. I was wondering why you chose to do the HCA versus, taking the assigned networks from Power.
Well, so yeah, so when you're trying to figure out, what are we going to call networks, so that we can look at their internal, connectivity, and we can look at their cross, network connectivity. So, how are we going to parcel up the brain into networks? And so one way to go is to use networks that people have, defined in the literature. So, there's the DMN, there's a, dorsal, attentional network. So, I mean, you could use networks that have been predefined in the literature. And so that's one way to go. And that's, I think, kind of what you're referring to. We decided, but there's controversy in that, right? People actually don't all agree on how to define those networks. They're defined in, actually rather different ways when you dig into them. And so we decided that we would look at what, our healthy control data show in terms of the clustering. And actually, if you look at them, there is a lot of similarity between some of them, as there should be, between these sort of previously established networks. So, we see the dorsal frontal parietal network. We see something that looks at least like what some people's DMNs look like, and so on. So, we were okay with this because we thought it revealed things that seemed pretty sensible. Mm-hmm. I mean, there are a lot of ways to go. Anybody who does network stuff knows all the decision points. Yeah, so that's a fine question. So we're looking at, so this is the this is the module or the component or the network where we're seeing these pre to post internal network changes. So it's the connectivity among these nodes that's changing from pre to post treatment. And it's interesting to kind of visualize that, how connected are these nodes to one another? So if you look on average in the control participants, each of these dots is, corresponds to a node, and you can see the connectivity among the nodes, in a healthy, when this module is healthy. And then this is the connectivity among those nodes before treatment. You can see how much sparser it is. That's why we saw that, this module has low, and not only this module, but many modules have this low internal connectivity prior to treatment. And it's nice to kind of see that visually. And hopefully you can see that post-treatment, you have an increasing density of the connectivity value. So it's becoming increasingly connected following treatment, which is what we saw. And it's starting to look more like the healthy control network. So you can see these changes in the degree. Remember, what we're looking at is not white matter connectivity, but we're looking at the changes in the degree to which the activity of these nodes is synchronized from before treatment to after treatment. And we're seeing this increasing synchronization of activity in these areas from before to after treatment. Okay, so to summarize this sort of level of analysis, I think it's really important to highlight that these within network changes that we're seeing, they're not evenly distributed across the brain. It's not like every network, is changing in this way, at least not under these circumstances. Not every network is changing in this way. In dysgraphia, this is the case that we're, the population that we're looking at, and we're looking at this background connectivity that is derived from a spelling task. We're seeing these changes in this particular ventral occipital temporal cortex region, and our interpretation, working interpretation, is that these local connectivity changes within this key node. We know this is a key node of the spelling network. They reflect, at least, the increased efficiency, of the spelling process. And maybe the fact that they're so localized accounts for the fact that the treatment results themselves were so localized. Remember, they're just improving in spelling. They're not improving in anything else. maybe we shouldn't be surprised that the network that's improving, is part of the spelling network, and that it's only that network. Not only, there are changes elsewhere in the brain, but that is really the, the star of the show in terms of within network changes. Okay. You're all with me still? Okay, so we're going to step one level up. Okay, and so now we're calling these 10 clusters, or clusters, or we're calling them networks or modules. Remember, I'm using those terms interchangeably. And we were looking at the connectivity within the modules. Now we're going to look at the connectivity between modules. Okay, and this gets me to what you wanted me to talk about, Laura, which was this notion of a network phenotype of stroke. So Siegel and colleagues came out with a paper, and Dorothy Sauer and all in a review followed up on it, and also referred to it in this way. Siegel et al. Had a very large group of subacute stroke individuals. They had a range of different deficit types. Actually, only a relatively small subset of them had language deficits, but that's okay. And they highlighted two results from a number of different analyses that they did. One result, sorry, did I say this right? Yeah. So one result was that there was higher connectivity in the patients and in the controls in terms of the connectivity between modules, between what they call modules. They define modules a little differently than we did, but it's still conceptually it's the same. There's still clumps of areas, that have, nodes internal to them. And so they evaluated the connectivity between the functional connectivity between them. And they found that overall there was higher than normal connectivity between them. So that the modules sort of communicating more than normal with each other. If you wish, they're less segregated than they would be in the normal brain, right? Because they're more communicating with one another and less segregated. And I see UN there. So UN, if by any chance I say something wrong, you need to put it in the chat. Okay. It can be, hard to keep track of the higher's and lower's and more's and less's. Okay. But this is what they said. Okay. And they also described lower functional connectivity between the hemispheres. Okay. So this gets to two levels, and I'll talk about the hemispheres next, but just to lay out what their network phenotype of stroke was this higher connectivity between modules, within a hemisphere. And they also have this cross hemisphere effect, which I'll talk about. That'll be the last thing I talk about. Okay, so I'm going to first talk about this higher within hemisphere functional connectivity. Okay. Okay, and so in a recent paper with UN, we replicated this stroke phenotype, and we were looking at chronic stroke. They had looked at acute stroke. There's some other differences in terms of what we did. So I guess it's, makes it, somewhat more reliable that we replicated. We'll start with that first characteristic of the higher connectivity between modules. Remember, we have these reference modules, and we're looking now not at how connected they are internally, but what their patterns of synchronicity look like across modules. So for any given module, if you look at all of its nodes and all of its connections with every other module, how synchronous is the firing of the nodes of module A with the nodes of all the other modules. Okay, that would be the cross network or the cross module functional connectivity. Okay, and so what we found was higher than normal cross network connectivity. You can see it both, it was true overall, and you can see it very significantly so in the left hemisphere, and numerically and approaching significance also in the right hemisphere. And you can see it graphically in these lovely images that UN created, where so connections that are in the blue part of the scale are connections that are weaker in the patients than in the healthy controls, and in the orange, yellow part, they're stronger. So there's two things to note: that sort of overall, there's more yellow orange than there is blue. So that's true. That's what creates a sort of overall higher intermodule connectivity. That's the yellow orange you can see, but there is green blue too, right? So it's not a simple story that all the modules are just more highly connected to one another, like end of story, right? It clearly is more complex than that. In fact, it's really quite complex. So there is that overall pattern, and maybe that's significant or not, that's hard to know. I mean, it's we need to still sort that out. It is certainly there overall, but clearly there's more complexity to that than that summary statistic would suggest. And so in this particular paper, what Yuan did was she evaluated the cross-network connections to see, to try to identify which ones are changing from pre to post treatment. Okay, a little more complicated, many steps involved, but that's basically, you know, what that analysis did. It was a classification analysis that was designed to identify which modules show significant changes in their cross-module connectivity. It's possible that the modules are colored differently in this schematic. I can't remember anymore. So if you remember the other schematic, don't get hung up on it. Just focus on the schematic that's here. Okay, they're different papers, so sometimes different color schemes get used. Okay, so that's what was done here. You know, we're just trying to understand. So given, you know, these modules, given that, you know, they, you know, we can evaluate the activity, you know, how synchronous the activity is across the modules, you know, given that we know that on, you know, on average, they're higher than healthy controls, what happens as a result of treatment? So let's try to identify which modules show these changes from before to after treatment. And what we found was that there were 3 modules that when you analyze their connections, so we're taking each module, we're looking at each module's connection with every other module. And we're saying which modules show changes in their connectivity with other modules from before to after treatment, okay? And so what we find is that there's 3 modules whose connections on average, you know, show a change from before to after treatment with other modules, you know, again, on average across the other modules. Okay, there's a left perisylvian module. Its connections with other modules are changing from before to after treatment. And the left and right dorsal prefrontal modules, each of them, their connections with other modules in the brain are changing from before to after treatment. Okay, so this is why it's cross-module connectivity. So what does that look like? So then we can say, okay, so overall it's changing, but, you know, is it changing more with some areas than with other areas, right? I mean, you know, it's changing overall, but again, you know, what does the distribution of those changes look like? And, you know, some changes will be increases in you know, they're more synchronized after treatment. Some will be less synchronized after treatment. So, you know, are we looking at increases in, you know, connectivity, decreases in connectivity, and how is it distributed across the brain? Okay. And so yes, so we have these 3 and we're asking that question. So for the left perisylvian module, I don't want to spend a lot of time on it, although I know many of us are interested in it. We, you know, probably could talk about it. But what this figure is showing you, so the yellow is showing you where the perisylvian module is. So it's the nodes in this module whose connections are being evaluated, right? So this module's nodes are being evaluated. And what the color is showing you, I didn't copy this very well, but what the color is showing you is the connections that increase from pre to post treatment. So their connectivity is increasing, and the pink shows you the ones where the connectivity is decreasing from pre to post treatment. And you can just kind of look at it. You can see there's like kind of pink in lots of places, and there's kind of green in lots of places, and it doesn't really jump out at you that there's certain, you know, areas where this, you know, where the, you know, where the nodes in this module are, you know, definitely increasing, or definitely, there doesn't seem to be a clear pattern. In fact, statistically, there isn't. You know, the nodes, there are significant changes. So these are significant changes, but there isn't a clear pattern. There's just lots of different kinds of changes that are coming from, you know, the nodes in this particular module. Okay, that doesn't mean they're not interesting. It just means that there's not, you know, a simple pattern. For the left and right dorsal prefrontal modules, however, there is a pretty clear and straightforward pattern to their connectivity changes. So you can see the pink and you can see the green. Remember, the pink are the connections that are decreasing in their connectivity. The green are the connections that are increasing in their connectivity. And if you just like eyeball it, you can see that there's a lot of pink lines going from the front of the brain to the back of the brain, okay? So these frontal areas are decreasing their connectivity with these posterior areas. That seems to be, you know, the eyeball analysis. And the green is not entirely, but mostly concentrated in sort of nearby frontal areas. That's what this, I mean, it's a glass brain, but that's, you know, your eyeball analysis, what it shows you. And when we actually, you know, evaluate the connections that show significant changes, or if you wish, the informative connections, you know, these things are hard again. So, but just to kind of walk you through it, these are the 10 clusters, right? These are the clusters. So for, you know, for the left dorsal prefrontal, the nodes in here, we're looking at all of its connections with these different with the other different components, like how, whether the connections are increasing or decreasing from pre to post treatment with each of these. And so what you have here, sorry, this one is the left dorsal prefrontal, this graph here, and this is for the right. Okay, they show overall very similar results. And what it's showing you though is the above 0 are increases in connectivity and below 0 are decreases in connectivity from pre to post treatment. Okay, and the colors are the colors of these modules. So it's the degree to which the connections of these nodes are increasing or decreasing with these other modules in the brain from before to after treatment. Again, above 0 are increases and below 0 are decreases from pre to post treatment. And so you can see, first of all, that eyeball, the green stuff that we're seeing over here is nicely green here. And you can see that these, the frontal areas connections with the dorsal parietal region are increasing from pre to post treatment. That's what the green that we're seeing here. And in fact, those are the most informative connections are all above 0. You can see it in the left hemisphere and you can also see it in the right hemisphere. Okay, in contrast, what you see is that these areas here, these posterior areas, which in this schematic are this turquoise and red, this kind of, no, sorry, not that. This turquoise and yellow orange here. Yeah, sorry, the turquoise and yellow orange colors. Okay, these posterior areas that include this ventral occipital temporal cortex, these areas are decreasing in their connectivity from pre to post treatment. Okay? And you see basically the same pattern, maybe not quite as striking in the left hemisphere, in the right hemisphere, sorry. And this is just highlighting this turquoise area, yeah, which includes that ventral occipital temporal cortex that we've referred to, so often in the previous results. So let me just summarize this in terms of, and we're almost done, guys. Between network connectivity changes, we see that treatment creates specific between network changes, right? It includes, these changes include decoupling of networks. So you see these frontal areas decoupling from the posterior areas, but you also see increased coupling. So it can go either way, right? You can see the decoupling from the posterior areas and you see the increased coupling with the dorsal parietal areas. What we suggest, so this is dysgraphia, right? And so we see this decoupling from these posterior, what we would call spelling areas, and this increased coupling from these frontal areas. Okay. Our interpretation is that perhaps with the increase in spelling efficiency that we've seen, remember we saw that this posterior area was internally becoming more connected, the representations were becoming more differentiated. Perhaps with increasing spelling efficiency, these frontal areas, may be less needed in spelling. And so there's sort of the frontal executive control is sort of decoupling from the posterior areas as a result of treatment with less executive functioning perhaps required. That's certainly, a speculation, but it, not unreasonable. Okay, and finally, and the shortest one because we have the least to say about it, maybe it's the most intriguing to me because it was the least expected from my point of view. I just didn't know anything about cross-hemispheric stuff. So the last sort of highest level analysis are the two hemispheres. We looked at homolog connectivity. Remember, Siegel had said that, across the stroke population, there was this lower between hemisphere connectivity, sort of across homologous areas, okay? And we find the same thing. And it was a very, very robust result, okay? Some things are a little more delicate, but this was just solid. Like, analyze it however you want. This is there, okay? So you can see that overall, this lower homotopic connectivity in the lesion group and the healthy control group, and you can see it also, the blue are the connections that are weaker in the lesion than the healthy controls, and you can see that this is, overwhelmingly blue. So most of these connections between homolog areas are lower in the patients than the controls. Of course, nothing is 100%, but it's very strong and very, it's very general. And so, is this cross-hemispheric connectivity, is it behaviorally relevant? I mean, we're finding it, but, does it, is it connected to the behavior of interest, in any way? And we find that it does. So the degree, I mean, this is where I'm most happy to get, feedback. The degree to which, for every individual, we can compute how connected the two hemispheres are, the homotopic areas of the two hemispheres are on average. So for every individual, we have a measure of how connected they are. For every individual, we know how severe their spelling deficit is, and what you see is a very strong relationship between these two between these two things. Okay. So how do they change with treatment? So with treatment, remember before treatment, they were low. And what we find with treatment, you see all these green lines, all those green lines are increases. So what we find is a generalized increase in homolog functional connectivity from before to after treatment. The vast majority of the changes that we see are increases. There's some decreases, you know, it's not 100, nothing is 100%, but it's very striking the degree to which the changes that we're seeing are increases, which of course is a normalization. You're going towards a more normal state. They were extremely low before treatment. They're becoming higher after treatment. And are they equally distributed across the brain, across all the homologs? And you can see that you know, here we're plotting the most informative connections, and you can see that all the modules do show these increases, but you know, some of them show some decreases too. And the ones that are really, you know, although it is sort of across the board, the ones where these increases are really, you know, standing out and are absolutely consistent are between, so this is between homologous areas now. So the dorsal prefrontal areas are increasing their connectivity tremendously, and also the dorsal parietal areas, again, with homolog, we're just looking at homologs here. They're increasing their connectivity with their homologs very much so from before to after treatment. Okay, so to summarize the interhemispheric stuff, we see this generalized increase in interhemispheric connectivity, especially for these homologous frontal parietal areas. And I don't know what the interpretation is, so I'd be happy to talk about that. Okay, I mean, I can imagine some things, but this is really, you know, not something that I feel like I have a good handle on. Okay, so just overall summary and discussion. Remember, we started off by looking at what behavioral recovery looks like. So we saw what that looks like. We saw how, you know, the spelling is improving, you know, how slowly a person is able to, you know, recover more and more and remember more and more of the letters of the word. So we know what that looks like. And what we've tried to do is to try to say, well, what does the brain changes look like? You know, we know what behavioral recovery look like. What does neural recovery look like in this case? And there are many things to look at in the brain, but what we've seen here is that recovery is happening. We are seeing signs of recovery at many different levels. We're seeing this local representational differentiation in the voxel to voxel relationships. We're seeing a within module change in the connectivity of the nodes within a module. We're seeing coupling, you know, increased coupling and decoupling of nodes across modules. And we're seeing these changes that are in terms of increased coupling across the hemispheres. So that's a summary of what we've seen. That's what it looks like from the brain point of view. So that's the voxels to hemisphere. And what are the lessons? I don't know, but certainly one of the lessons is that recovery involves network changes at multiple levels. That maybe is not surprising, but I think it's interesting to sort of lay that out. It's also, I think, important to note when people work on networks, sometimes they, you know, tend to, you know, talk about networks very globally or in terms of whole brain networks. But we're seeing here is that not all recovery related change, not at all, is really global or very widely distributed. You have local, you know, changes at all these levels. You have local changes. It's not uniform changes across the brain. And there are, you know, any number of questions, but certainly the question that I'm really that really is, you know, a burning question for me, is, well, what is the relationship between the changes at these different levels? Like, are these just different things that are happening? You know, they're all relevant to recovery, but they're just like different things being fixed, you know, or does one drive the other? Maybe it all starts at the bottom. Maybe, you know, the local differentiation just cascades, you know, and creates all these changes throughout the brain, or not. I don't know, but that's certainly what, you know, I would love to understand. And then finally, you know, is there any kind of clinical relevance or application? You know, I don't, you know, but certainly I think it's important for us to always have this in mind. You know, is there some way in which, you know, this is actually relevant, you know, to clinic either now, or can we see, you know, a direction in the future for that? Is it useful for predicting response to treatment? It's only useful if it's more useful than something that's easier to do, right? If you can predict response to treatment from some easy behavioral task, why are you going to go through and do some like crazy set of, you know, network analyses, right? So, you know, if it's going to, you know, be useful for, you know, in that regard, I think it needs to prove itself to be more useful than something that's easier to do. But maybe it could provide guidance in identifying targets for stimulation and so on. So hopefully there would be some utility to this, but right now it's really kind of at the basic science level. And I just want to acknowledge the great team, all the great patients, and thank you all for listening.
Wonderful, Brenda. Applause. Yay. I had some.
I wasn't entirely crazy in my time. Oh my,
your timing is great.
I am such a bad, everybody who knows me knows how this is like my major source of stress is like, how long is something gonna last?
Perfect. There were quite a number of questions. And
should I stop the share or what should I do about that, Laurel?
Sorry, you may want to keep it because
okay,
some of the people may want to go back. So
okay, no
I'll just call on a couple people who had questions during the talk. Some of them you did answer. I know Peter Turkletom. Hello, Peter. Welcome. Had some questions for you.
Hi, Brenda. How are you?
So why don't we start with that?
Okay, first, Brenda, I think you know how much I love this work. I think it's really great.
Well, thank you.
I did want to ask, just for the connectivity stuff, how much do you think that depends on having done the connectivity in the context of the spelling task?
Yeah.
Like, do you think you'd do the same thing if you'd done it just in resting state?
So I hope UN is listening carefully to your question. Because I mean, you know, there's always you have a list of priority projects, right? And this isn't at the top of our priority list, but on our list is we have resting state data from all these people. So, you know, it's really, I mean, nothing is straightforward, but we have the data that will allow us to see whether or not we get the same kind of results when we just use resting state. So are these kinds of changes evident just at rest when people aren't doing anything in the scanner and we're just looking at the rest? Or is it driven by the fact that this measure, even though people have, you know, other people have said is very, you know, the main, you know, when people sort of analyze the components of this background connectivity signal, they said the main component is, or, you know, you see this great similarity with what you see at rest. So they, you know, done that in other contexts. And then there's like a lesser component that has to do with, you know, processes that are shared across, you know, like shared components across tasks. And then the sort of the smallest component is the particular task where you collect the data. But there is a component that's related to the task that was used when the data were collected. And so I don't know the answer to that question. It could be that we're able to, you know, it could be that we're only able to really see this kind of structure because there is still this task element, you know, that's in the signal. And so we just need to do this. I mean, what we want to do, what's sort of on that list, is to do exactly the same analyses with exactly the same participants. And I think that's the way we'll get, you know, I think we need to know this, because if resting state will give us the same results, it's way easier to collect resting state data than to collect task-based data. And so we really need to know the answer to that question.
There are other questions, but since it's so related to Peter's, I wanted you to clarify about the increase in within network connectivity you saw in the patients, even outside of, to forget the treatment.
Wait, I can't hear you.
Sorry, the within network connectivity was increased in the patients, right?
Yeah.
And that finding is not typical of resting state studies in patients. If and I'm not the biggest expert, many people here who know more than I, but certainly in other domains, I know we see decreased connectivity within hemisphere in patients who've had stroke. So I wondered if it had something to do with the fact that you used a task.
So are you talking about the within a module or across the modules within a hemisphere?
Across the modules within a hemisphere.
So across the modules within the hemisphere, that's where, you know, we see coupling and decoupling. So that's these kinds of results, right? So we see that, let's say for that.
But so many of them were increased.
You know, in the stroke, yeah, the overall pattern in the, oh yeah, so pre-treatment, the average was overall higher than normal, yes.
And that might be related to the fact that you had a task in the scanner.
Oh, you're saying that normally if
you're doing resting state,
you don't see that
reduced.
Oh, okay, yeah. So yeah, you're more familiar with that than I am. Yeah. So that
someone else correct me here who knows, but that's what I've seen in the resting state literature.
Yeah.
So that will be of interest, I think.
Yeah.
Okay.
Yeah.
Thanks.
Yeah.
The next person.
We'll look at that.
Okay, I want to clarify actually about that question. About the between network connectivity in the left hemisphere, which is the lesion hemisphere in our patients, right?
Yeah, this.
So yeah, so we have aphasia. So they have like an MCA style lesion there. So actually, I would say the pattern is a bit complicated.
Yes.
Than the original phenotype paper, which they look at, I would say, a broad range of participants. So I think it is true that if you look into it, the pattern is more complicated in terms of the within. I think we have like maybe another figure, but Brenda didn't put it here. But I think the pattern, especially in the left hemisphere, is more complicated in aphasic population than the original. Yeah.
In terms of the stroke phenotype, so the claim was the stroke phenotype was this higher overall within hemisphere, across, module connectivity. And we do find that overall. But as Yuen said, I mean, that's one of these results that I would say, like the cross hemisphere results, they're rock solid. They're not going to go away. The within hemisphere, patterns, I think are, they're complex. They're not just, it's not the case for all the modules. I think that that's going to vary depending on, where the primary lesion might be. So I feel like those are, more variable results. But it also could be that there's a different, that they pattern differently in resting state versus this task state. And I think that we need to understand that.
Right. We have a few questions in line here. John, then Paigey, and then Sharon. John White, do you want to?
Hi, thanks, Brenda. Really enjoyed your talk. Yeah, I wondered if you could reflect a little bit on the finding that that lesions remotely seem to cause unlearning in an undamaged area. So I understand your notion about how the differentiation occurs during learning, but how do you think about how remote lesions somehow undo that learning?
Yeah, so that's a great question, right? So I think that what you're, let me just get this. Let me see. Sorry, it's not always the most efficient way to find things, I know, guys. Okay, so if we think that these are sort of the brain areas that are involved in spelling, right? These multiple brain areas are involved in spelling, and they, participate in the sort of various cognitive functions in some more or less complicated or not complicated way. What you're, I think, asking is, okay, so we're finding these changes in this undamaged part of the brain, right? So these are changes in the undamaged part of the brain. People had lesions, over, for the most part, over in here, right? And we're finding, I think that what you're really referring to is that we're finding that even before treatment, right? If you just look at these people, right, before we've even done any treatment, and you look at their spelling, severity of their spelling deficits, you find that the severity of their spelling deficits, is correlated with the quality of the representations in this part of the brain. I think what you're asking is, how did damage all the way over here impact the quality of representations in this undamaged part of the brain, right?
Yeah, and I guess it also has implications for how you think about what is represented there, because if the knowledge of spelling is represented there, then where did it go? But presumably it's something more complex that's instantiated there.
That's right. And so that's why I always like, when I say representation of spelling or access to them, I often say those two things, because because what I actually think, I think that if you damage this part of the brain, you can certainly directly disrupt these representations. But I think that probably what's happening is that access to these representations is controlled by these frontal areas. Okay, I think this is involved in lexical selection. We have some other reasons to think that. And so I think that these disruptions here are creating difficulties in, selecting, distinguishing, navigating through the knowledge that's stored, probably in this part of the brain. And so I think it's the damage in this area that's impacting the ability of this area to function, as it should. And that by strengthening these representations, and maybe it's degrading them because, there's prolonged inability to actually retrieve them, you're having trouble retrieving them. And so the system starts to deteriorate because of these failures in other parts of the system, functionally starts to deteriorate. these areas start to become decoupled from each other because of all the failure that this part of the brain, even though itself isn't damaged, it is failing, you know, as someone is trying to spell. And so you're not able to keep exercising that area and refining it as you do when you're, you know, successfully spelling. And so you're creating sort of noise in this area because of failure somewhere else. That's the best I can do. I mean, I don't think it's a crazy idea, but what do you think?
Well, yeah, I mean, I guess the other way that I was thinking about it is that what is instantiated there is kind of where to get the information. I mean, you know, the connections to the sort of raw data that allow you to spell, and that as those source materials get lost, you know, you don't have the information to make the differentiation. And then presumably what happens during learning is somehow other parts of the brain are able to make some discriminations that now can get connected here and re-differentiate. That's just sort of a slightly different way of thinking about it, I guess.
Yeah, maybe it's kind of the reverse. You know, this is the area that, you know, allows you to go find the information. When that information is all messed up, you know, it no longer knows how to find it. But if you can, you know, strengthen it, then it's sort of able to, you know, have, you know, cleaner instructions to those areas. Yes, it could be. I mean, we really have, you know, no way of, at least I don't know how to distinguish between these things. That's why I try to be a little vague. I mean, it has something to do with these lexical representations, how they're represented and processed, retrieved, selected. Yeah.
Yeah. Thanks a lot.
Yeah, no, thank you.
I know a couple more questions. I know Paige, you had a question.
Yeah, actually mine just really dovetails with what John was asking. I was thinking very much the same kind of thing, and thinking about that, you know, we think about, okay, their orthographic knowledge or access to it is impaired, but because of these lesions that are outside that area, okay, and you make these changes, so yes, you could be changing that region, but much more so, I'd be thinking that you're changing the residual network that accesses it. And then thinking about your results, though, being so robust in that posterior temporal-occipital area, is that because it's the final common pathway?
Could be.
And it's not damaged.
Absolutely, it could be. You know, I mean, I think that's why, you know, and every time I said like interpretation, right? Because I think it's really difficult. And so, in fact, if we look, Paige, if we just look at mean bold, if we go back to that very first result in terms of looking at mean bold changes from pre to post, those are really concentrated out of bilaterally angular gyrus. So maybe that's where the, you know, the real representational changes are taking place, you know, and they are then, you know, in some way, you know, influencing, you know, these other, you know, these other kinds of processes and representations. I mean, this is really hard, right?
But I also like the complementary findings that you had relative to dorsal parietal, because effort and working on it. Yeah.
You mean the decoupling from those frontal? Yeah. I thought that was really fascinating to find that. Yeah. Yeah. I mean, I feel like we have this puzzle. We have these puzzle pieces, right? We have a very difficult puzzle, you know, not like a 5,000. Those of you who like jigsaw puzzles, and we have a few pieces. We're trying to figure out what is this all about. But yeah, I mean, it's a great point. I mean, it dovetails very well with John's point. Yeah.
Thanks.
Sharon, you had a question, and then I think we have to wrap up unless there's any other burning questions.
Yeah,
go ahead, Sharon.
And anyone can cut me if they'd like. But I'm just interested to know, Brenda, what your interpretation is of the increased coupling in the dorsolateral prefrontal cortex. So we're talking about the decoupling of frontal with the posterior temporal, and I'm just curious what you think that other pattern was about.
Yeah, that's where I mean, I need help everywhere, but honestly, I think that's where I need the most help. You know, I mean, it's very robust. So the prefrontal area, so within hemispheres, so there's two results there. So within hemispheres, the dorsal prefrontal, you know, nodes increase their coupling with these dorsal parietal nodes, and that happens, you know, within each hemisphere. So those two areas, you know, within each hemisphere become more coupled, and then each of those areas becomes more coupled with their homologous region across hemispheres. You know, so they're internally coupling, you know, to each other, and then they're cross hemispherically coupling. And do you have any thoughts about that? I mean, I'm not, you know, I don't know that much about this part of the brain, so.
I'm struggling with it, and so I was I was hoping that someone smarter than I, would have
No, but
would have the answer.
No, but there may be people here that, you know, just know more about this part of the brain too. I don't know. Does anybody have any thoughts and suggestions? Anybody illuminating insights? Yeah, I don't know. So I just don't know, right? I mean, you know, I don't know. I don't know.
Does it have something to do with, I don't know, with self-monitoring or some response to the learning?
Yeah.
Yeah.
So, yeah. So could it, you know, it doesn't, you know, again, so this goes back to the selectivity of the treatment effect. So we didn't see, you know, broad, you know, treatment changes, you know, it wasn't like treatment was changing all kinds of things, right? It was changing spelling and it wasn't changing, at least it wasn't changing the other tasks that we looked at, which were really, you know, language tasks, you know, auditory comprehension, you know, naming, I don't know, we looked at, you know, a few things. So it wasn't changing those tasks, but maybe the treatment was changing something else, like, you know, some general monitoring function, like you're saying, you know, and so being in this kind of treatment environment, you know, maybe it changed this general monitoring function. It wasn't, you know, it's decoupling from the, you know, posterior part of the brain. So it's not that it was needed so much anymore for the spelling, but, you know, this is a change that's happening, maybe a side effect of the treatment that we didn't measure. You know, we didn't do a comprehensive executive functioning battery before and after treatment. We have a couple of tasks. We didn't see, you know, any change in that, but we didn't really, you know, we didn't do a Wisconsin. We didn't do this and that, you know, so it's possible that, you know, that training had these broader executive consequences that we just didn't measure behaviorally, you know, because we actually didn't set out to, you know, I mean, we only just sort of tapped at it. We didn't find anything, but we didn't really dig into that. So that's probably the best I can do, but I know it's not that satisfying.
No, it's great. Thank you.
Yeah, you're welcome. Yeah.
Okay, it's after 5 now. So I think we should wrap up, and this was fabulous and so many interesting questions and things to talk about. Thank you all very much for joining us.
Well, thank you so much for having me. It was
great.
I really enjoyed it. It's so many levels. It's great to see all these faces, you know, and it's also really great to share this work. You know, we've been working hard on sort of our network kinds of analyses the last few years, and it's, you know, nice to try to bring it together and try to start thinking about, you know, how this might all fit together and what it all might mean. And your questions are really great. So thank you.